Hello, everyone. Welcome back to Brainwaves. Today on Brainwaves, I welcome back June Lee, PhD. Dr. Lee is a assistant professor of human genetics at University of Michigan, and he's also an IMRO Johnson & Johnson Translational Rising Star Research Award winner from 2011. Dr. Lee has appeared on Brainwaves once before, at which time he discussed his lab's highly focused approach to understanding the roots of bipolar disorder. And today he's going to tell us about some more progress he's made there and in another area of understanding the roots of mood disorder, specifically in understanding the roots of depression. Since I talked to June last, he and his lab have published a great paper talking about the evidence they found for why people with major depression often don't sleep very well, why they have a disrupted sleeping and waking cycle, and excuse me, as it turns out, the sleep issues may have something to do with the expression of their genes. So, and this discovery may point to some leads for new therapies. So June, thank you for being back with us on Brainwaves today.
You're welcome, Brandon.
Well, great. As I mentioned a second ago, your lab has discovered what seems to be a very evident gene expression signature for the basis
Uh-huh.
for the disrupted cycle of sleeping and waking that people with major depressive disorder so often experience. Now,
Right.
yeah, and the paper you published was really well received on this. And what makes you suspect that these sleep issues have a basis in gene activity?
Um, First of all, I wouldn't call this my study. It is a large collaboration with a lot of seasoned researchers working together across many universities. Uh, so, in this study, we looked at gene expressions in postmortem brain tissues of donors who agreed to participate in research. So when they die, their brain tissues are made available for us to look at the gene expression signature. Our original goal is to compare between normal controls and those who suffered mental disorders. But along the way, we realized we need to figure out what other factors could be influencing gene expression. So one of which is the hour of death during the day. So in this analysis, we figured out the gene expression which reports gene activity in multiple brain regions actually did show cyclic patterns around the 24-hour clock. So this was suspected before, but never really proven or settled. The reason is the human brain tissue is very difficult to access. It's impossible to, say, follow the same person around the clock, take one sample per hour. So in our case, we put each individual at a single point, which is hour of death. So across several dozen people, they populate around the clock, but everyone only contributed one data point. But because the circadian pattern is so robust, we were lucky enough to actually see it. See many genes are actually cycling. So this settled the old question whether they are cyclic gene activity patterns in the brain. So now we know they are. This is previously only known for animal brains. And for humans, you can only show that for skin cells or blood cells, those easily accessible cells.
Okay, so well, that's fantastic. You've actually seen this in the genes in the brain. So now that you've made this discovery, well, first you found that there was a cyclic pattern in people with who are healthy controls, but then
Mm-hmm.
people who have major depressive disorder have a different signature.
Right. So when we talk about gene activities, the microarray-based research gives you many thousands of genes to begin with. So if you find several hundred that are cycling, they actually cycle at a different phase. So some genes might be reaching the highest point in the morning, but drop to the lowest in the afternoon. The next gene might peak at noon and then hit the valley at the sunset time. So once you have this pattern, you learn it in the healthy people, you can apply it as a predictor. So next time, if there is a new sample coming in, we can look across those most informative genes and then look at it high, low, high, low, and using it almost like a barcode to say, hey, this particular string of high, low is an afternoon pattern or it's an evening pattern. So when we apply this predictor to the bipolar disorder patient, we found their brain is carrying what we call a faulty signature. Some of them apparently is cycling at the wrong time, as if they are living in a different time zone. Others have just uncoupled regulation, as if they had poor sleep, not only just sleeping at the wrong time. So this is a revelation that it confirmed people's suspicion over the years that sleep disruption is related to mood disorder, even though it's difficult to really sort out which is the first, which is the consequence. But for us, we are now confirming this particular correlation at the cellular level and also highlighting the genes that might be involved in the process.
Wow. All right, that's really exciting. And now that you've discovered this correlation between uh, the unusual patterns of sleeping and waking people with depression tend to experience, and bipolar disorder also tend to experience, and then this expression pattern in their genes. What can you do with this discovery for potentially helping patients with these diseases?
Right, right. That's a good question. So before we made this discovery, it's a lot have already been known about the relationship between sleeping patterns and mood disorders. And then clinicians have always thought about how to put the patients who have mood disorder through sleep therapy or through light therapy. One example is the so-called seasonal affective disorder, the winter blues, people who have mild depression in winter time when the daylight hours are short. So if you can do the light therapy by adjusting the brightness, the quality, and the length of the light, people can restore better sleeping patterns, and then actually that can lighten the mood. So all of these are known on the behavior level. And what we are contributing is trying to figure out how this is happening in the brain, which brain region are involved, and then which genes are involved. And then when we say the depressed patient have the disrupted pattern, which genes are disrupted? Do they cycle at the wrong time or do they cycle at the wrong magnitude? Or which pairs of genes used to be in phase with each other and now become out of phase? So these molecular details are newly discovered through our study. So we hope it provides a more solid foundation, a knowledge base for future research.
Okay, that makes sense. Would you care to speculate that maybe someday the knowledge of what genes are involved in disrupted sleeping and waking patterns could be used for perhaps developing pharmaceutical or gene expression therapies for depression?
Right. I believe this will happen. The question is how soon it will happen and how do we get there?
Sure.
We all know the brain science is difficult simply because the brain is such a complex system. It's far more complex than other organs. You might say for liver it will just do what liver cells do and then just more of it. But brain will do what neurons do, what much more than that. So it's once you know the genes, there is a long way to figure out how the gene is actually disrupting the network behavior of the brain. And then in our case, since we know the specific genes, it gave some functional leads to do reductionist research to actually experimentally tease apart different components of the network, either in cell models or in animal models, and then to observe network behavior. The circadian pattern is actually one part of this puzzle. We know mood disorder is actually more complex than circadian regulation. And then understanding how the circadian regulation are disrupted can provide essential clues on how mood regulation might be disrupted.
Okay. Very good. Very good. And then another really good area that you've been researching is the candidate genes for disruption, candidate genes for bipolar disorder susceptibility in general.
Right.
And that was your original proposal for the Emerald Rising Star grant. So how is that research going? I understand you discovered a
Right.
few new genes for susceptibility for bipolar disorder?
Right. Just a little background. What we propose to do is to sequence the coding portions of the genome for individuals who are affected by bipolar disorder. But not just any individual, but choosing the samples from what we call the multiplex families, that are large families with multiple affected across multiple generations. So what we wanted to do is to, by way of sequencing, identify all the functionally damaging DNA variants and ask which ones are rare in the general population but shared by the affected individuals. So the proposal is to sequence 90 individuals. We have now finished most of it. We genotyped all 90. We finished sequencing on 82. So we have information on 29 out of the 34 families we proposed. So we have carried out a very detailed annotation of the variant, trying to figure out what are the predicted function of the discovered DNA variation. So this allowed us to filter down a short list of 500 variants distributed across those 30 or so families. And then within each family, you can further ask which ones are shared by the affected individuals. And then some families, depending on the family, some we have a long list, say 20 variants that are still waiting to be filtered down further. For two of other families, we found one or two variants left. And they actually, in two cases, we found the variant hit on the genes that previously known to be involved in psychiatric disorders. There was one reported for OCD, the obsessive compulsive disorder. Another family had two variants, one reported for schizophrenia and the other reported for ADHD. So these are interesting leads. It didn't really prove the functional explanation yet, but I think this is really hopeful because it allowed us to go further to ask how about the unaffected in those families. We can do more extensive variant screening in the family members to strengthen the case that the variant we discovered are indeed segregating with the disease. And then we can also further aggregate data with all the other concurrent studies to see if there could be other variants on the same genes or other disruptions on the same pathway that could be observed in other bipolar families.
Okay, great. Well, you're making incredible progress there, and at the very least, it underscores that that bipolar disorder and other mood disorders are biological disorders, which is important for people to understand.
Right.
Yeah. So thank you, June, for the work that you do,
Yeah.
and thanks again for picking on brainwaves. But before you go, I want to just mention to people that you're going to be at the Music Festival for Mental Health on September 7th. And so viewers, if you'd like to meet June and ask him questions about his research, he will be there. June, are you looking forward to meeting some guests at the festival?
Yeah, likewise, I look forward to seeing you and the other nice other colleagues and your other individuals that you have invited to the festival. Yeah, it's always a pleasure to meet with them.
Well, good. And then if people want to ask you some questions right now on brainwaves, are you ready to field a few questions?
Oh yes, absolutely.
Okay, okay, good. All right, thanks, June, again for appearing on brainwaves, and I hope to talk to you soon.
Yeah, you're welcome. It's a pleasure talking to you. Okay,
You too. Take care.
okay, bye bye.
Bye.